here we go. Looking forward to your presentation.
Many thanks for the introduction, and many thanks for the invitation and being here. So good afternoon, everybody, and Happy New Year too. So today I would like to share some perspective about how we can investigate the relationship between structure and function in the brain. And communication mechanism in brain network. So what I mean by communication, we all know that neuroimaging techniques allow us to probe a different dimension of the brain. From one side, we can probe the structure of the brain and the white matter connection between different brain regions that we can often represent in the form of a network where nodes represent different gray matter regions and link or edges represent the white matter bundle. On the other side, we can record neural activity-related signals so that we can ascribe to each one of those brain network nodes a time series representing or related to neural activity. Those time series are not independent, but it happened that they are organized into functional states or pattern of activity synchronization that sometimes we can also represent in the form of a connectivity matrix, where here entries represent some statistical relationship between those nodal time series. Now, in this framework, we can see the structural brain network, so the white matter wiring, as a skeleton that constrains the flow of information between the different parts of the brain. And we can investigate a communication pattern between different brain areas. So those flow of information between different brain areas through the underlying structural connection. Yet the disjoint assessment of brain structure and function does not directly reveal those communication patterns, which remain a sort of hidden dimension of the brain. And so that the relationship between brain structure and function which emerge on top of this structure is still not completely clear. So there are several methods that have been proposed in the last few years to investigate the relationship between the structure and function in the brain. We have both model-based method and data-driven approach. From one extreme of the model-based method, we have biophysical and computational models which reach the highest level of detail in modeling brain activity. So there, each brain network node or cortical region is represented as a neural mass, and then those neural mass are coupled according to some neurodynamical scheme through the underlying structural connectivity. And these produce a synthetic signal that then can be compared to empirical data. For example, we can build synthetic functional connectivity matrices and fit them to empirical data to estimate the parameter of the model. Then for a more network theory perspective, it's possible to investigate different communication models where we model a sort of propagation of information from a source to a target node according to some propagation rules. And this has been very nicely reviewed by Caio Seguin in the lecture in December, which I really enjoyed. So from a certain communication model, we can compute a sort of communication efficiency. So this is a measure based on some navigational rules on the structural connectivity only. And then we can compare that to empirical functional connectivity data. Then finally, there are different data-driven approach, mainly based on correlation. So from simple univariate correlation between structural and functional connectivity strength to multivariate correlation analysis, we show some degree of correlation between these two dimensions. So I think that one interesting aspect to consider when looking at these different approaches is that the functional information is typically used a posteriori in a way to compare different models, to validate different models, or to fit the parameter of the model. But it's not directly, let's say, it's not directly embedded into the communication modeling itself. So a perspective that I would like to give you today in this talk is to explore a different way to explicitly integrate the functional and structural data in brain communication model, or in brain communication approaches, both from a data-driven perspective, so introducing you some elements of graph signal processing methods, and from a model-based perspective by integrating or enriching communication models defined on the structural connectome with functional data. So using information theory elements. And from a more neuroscientific perspective, the question I would like to tackle is that the heterogeneity of the structure-function relationship in the brain across multiple levels. So is the structure-function relationship homogeneous across different cortical regions, or different cortical region might be associated with different communication paradigms? And then is this relationship similar across individual or specific to individual subjects in a group? And then even zooming out further, are these communication paradigms preserved across evolution or not? Okay, so let me start from the first data-driven approach for structure and function integration. So I think a natural way to look at functional recording is to see them as signals defined on top of the structural graph. So we are more used to look at signal defined on a regular domain, such as a time series defined on the temporal axis. But in a similar way, we can also define a signal on an irregular domain as a graph. So in this scenario, we can associate one value to each node. So basically, a signal on the graph will be just one specific time point of the fMRI recording, so that we have one value per node. And then we would like to answer a different kind of question. So for example, can we quantify the amount of structure-function coupling or alignment? What I mean with coupling, a functional activity pattern will be coupled with the underlying structural graph if it has a similar signal and connected node. It will be decoupled from the underlying structural graph if we'll have different signal values at the connected node. So in this example, for example, R and B will be coupled in a way, similar value at connected node, while B and C will be decoupled in a way, different value at connected node. And then in the vein of exploring the heterogeneity of the structure-function relationship across the cortical mantle, can we define some nodal measure of structure-function coupling? So to answer this question, one possibility is to extend the tools of the classical signal processing domain to the analysis of signal defined on top of graph. Now, the most fundamental tool for signal analysis is probably the Fourier transform and the Fourier analysis, which you all probably know. But just to recap, the idea here is to decompose a signal into a weighted sum of harmonics, where here the harmonics are sinusoid. We have low frequency harmonics with similar value across neighboring time point, and high frequency harmonics with different values at neighboring time points. And so by getting the Fourier transform of a signal, for example, here a time series, we can compute the spectral coefficient, which tell us the energy contribution of the different harmonics to the signal, here shown in the energy spectral density graph, right? So this signal will have a lot of low frequency component. Now, once we have the spectrum of a signal, we can perform several operations. For example, we might be interested in quantify or select just the low frequency of the signal and reconstruct only the low frequency of the signal. So the Fourier transform is an invertible operation. So given a spectrum, we can get back a signal or a part of a signal after a filtering procedure. And similarly, we can, for example, selecting only the high frequency part of a signal through, for example, an ideal high pass filter, right? Now, how can we extend this analytical approach to a signal defined on a graph? Well, let me reinterpret a temporal time series as a signal defined on a regular graph. So here each node will be a time point, and each node is connected to the previous node and to the following node, right? So we can describe this graph with a connectivity matrix or adjacency matrix that just describe the pattern of connectivity of the graph. And then we can complement this with the degree matrix. So this is just simply a diagonal matrix where on the diagonal we have the number of connection of each node. So in this case, each node is just 2 connections, right? So all value on the diagonal are 2. And then let me introduce a last element, which is the graph Laplacian, which is simply the element-wise subtraction of these 2 metrics, the degree and the adjacency matrix. Now, it turned out that the eigen decomposition of this Laplacian matrix define the Fourier transform of the graph. So the eigen decomposition of the matrix allowed to decompose the matrix into a series of eigenvectors U associated with eigenvalues lambda, where we can order the eigenvalues from the smallest to the largest one. Now let's see how this eigenvector of the regular graph look like, where we see that the eigenvector corresponding to the smallest eigenvalues indeed correspond to smooth variation over the line graph. So indeed correspond to a sinusoid. And then as soon as we move on with eigenvector associated to larger eigenvalues, we see appearing sinusoidal pattern of higher frequency, and so on for larger eigenvalue. So indeed, we can interpret those eigenvector as the graph harmonics, similar to the Fourier analysis in the temporal domain, and eigenvalues as graph frequency. And indeed, the eigencomposition of the Laplacian of this regular cycle graph is equivalent to a discrete Fourier transform of the similar signal. And we can define the Fourier transform of the signal X defined on the graph as the decomposition of this signal in terms of the contribution of the different graph harmonics, which are the eigenvector of the graph Laplacian. Again, the graph Fourier transform is invertible. So from the spectrum to the spectral coefficient of the signal, we can go back to the signal itself. So now I showed you the harmonics of a very simple graph, so the regular graph, but what happened with a more interesting graph? What happened with the brain graph? So here we have a structural brain network derived from diffusion-weighted MRI data. That we can eigendecompose, so we can compute the Laplacian and do the eigendecomposition of the Laplacian, and we will get those eigenvalues, which are here ordered for the smallest to the largest. And here I'm plotting some of the harmonics or the eigenvector associated with those eigenvalues. We see that those different structural harmonics can be interpreted like, let's say, building block of the structural connectivity networks and encode the different features of the structural connectivity pattern. For example, for lower frequency, for smaller eigenvalues, we have some geometric constraint of the structural connectivity pattern. Here you see we have a symmetry between the left and the right hemispheres, and then we have a posterior to anterior gradient. And then for larger eigenvalues, so for a larger higher frequency, we start to see more complex pattern appearing that encode some more intricate features of the structural network. So now the idea of the graph signal processing approach is, can we take a pattern of functional activity defined at the node of a network and reinterpret or decompose them in terms of these structural harmonics, or in terms of the building block of the structural graph. Here it's what happened when we take the graph Fourier transform of a resting state fMRI recording. So here, the gray area represents the different time points across the recording, because again, the graph signals is just the association of one value per node. So it will correspond to a single time point. And here what we can see is that the functional activity pattern are naturally smooth or naturally aligned with the underlying structural connectivity substrate. So here we see this nice power law relationship in this energy spectral density with a strong contribution of the lowest frequency. Yet we can think of applying a similar instrument as the filtering that I showed you before in the temporal domain to the graph domain. So what we could do is to quantify the contribution of the high frequency of the signal. So this would represent the component of the functional pattern which are decoupled, let's say, from the underlying structural network. And similarly, with an ideal low-pass filter, we can also select the part of the functional activity pattern which is coupled with the underlying structural network. Now, we can do different things with this quantity. One thing we can do is to define a local measure of structure-function relationship, or a local measure of structure-function coupling, by taking the norm in time of the signal decoupled from structure divided by the norm in time of the signal of that node coupled with the underlying structure. And so that we end up for each brain region with an index that tells us the amount of coupling or decoupling of function with structure locally at the specific brain location. On the other side, another thing we can do is to rebuild the functional connectivity pattern based only on the coupled or on the coupled functional activity pattern. So we can take the wall signal, compute the Fourier transform, consider only, let's say, the low frequency, reconstruct the low frequency part of the signal, and compute a coupled version of the functional connectivity matrix. And similar for the decoupled part of the functional connectivity matrix. Okay, so now that we have built all this machinery, what we can learn for this kind of analysis? The first thing we can learn is that indeed the relationship and the level of alignment of functional pattern with the underlying structural network is not homogeneous across the cortex, but vary from region to region. So here on the left, we see the wall cortical gradient of the structural decoupling index, and we see that region associated with lower order functions, such as the visual cortex and the somatomotor cortex, tends to have activity which is coupled with the underlying structural network. On the other side, the region which are associated with higher order function, cognitive function, present functional pattern with a strongest decoupled component from the underlying structural connectivity network. A second message here is that the functional activity pattern decoupled from the underlying structural network are relevant for inter-individual variability. In particular, they are specific to individual subjects and relate to behavior across different subjects. So to see this, we took the coupled and decoupled part of the functional activity pattern and built, as I was planning before, a coupled functional connectivity matrix. Here is ordered according to the classical resting state network. So you see strong functional connectivity within the resting state network. And the decoupled functional connectivity matrix. Here you see lower functional connectivity within the network, but more functional connectivity in a way between different networks. Then what we can do is to analyze how much this quantity are specific to individual subject. So to do this, we can play, let's say, the guess who game, where, so these are not functional connectivity matrices. These are scan-rescan correlation metrics between different subjects. So here row and column are subjects. And here we see the similarity between functional connectivity of subjects in the scan, every scan setting. And we compute these from classical functional connectivity matrices or from the decoupled part of those functional connectivity metrics. And what we can see is that the success rate in subject identification, so how much I can identify a subject just based on its functional connectivity data, is much larger if we look at the decoupled functional connectivity pattern than if we look at the full data functional connectivity information. And this is true also if we look at different settings when we try to identify a subject from a certain task, for example, in the resting state or in some performing some language or motor or other kind of task in the scanner, from data of the same subject performing another task in the scanner, again showing the relevance of the decoupled functional connectivity pattern. And in the same vein, the coupled functional connectivity pattern in a multivariate correlation analysis, in a multivariable correlation analysis with a cognitive profile of subjects. This is specifically is an analysis on the 100 subjects of the Human Connectome project. We see that the decoupled functional connectivity pattern offer or explain a larger amount of interindividual variation of cognitive traits. So this by embedding structural and functional data within a graph signal processing framework, we saw that the relationship between brain structure and function is heterogeneous across different brain regions, and this might suggest the presence of different communication protocols associated to different brain systems. And then we also saw the relevance of the decoupled part of functional activity pattern for subject specificity and for behavior prediction. I wanted to show also you this graph signal processing framework because here we're applying this to brain structure function data, but of course this is a general framework that can be extended to other kind of data defined on a complex or connectivity domain, let's say. So now we have investigated this coupled and decoupled functional activity pattern, but how can we interpret them? Okay, and this is just really a personal interpretation, so there is not a real answer or a clear answer to this. But to me, coupled the functional connectivity pattern or a functional activity pattern coupled with the structural domain might represent a pattern of synchronization that emerge from more direct communication, for example, supported by direct connection. On the other side, pattern of brain activity which are decoupled from structure might emerge from more complex mechanism or paradigm of brain communication. For example, through polysynaptic communication or to other kind of mechanism like integration of information, like integration of information within a cluster or more complex structure. So to untangle this different possibility, I think it's necessary to turn from a data-driven approach, like the graph signal processing, to a more model-based approach. And here I would like to share with you a second work that have been done in the last year, again, in trying to integrate the structural and functional information in the same model. So we decided to start with a very simple model of brain communication, which is that the two distant regions in the brain, let's say a source X and a target Z, can exchange information through indirect interaction along a polysynaptic structural path in the network. We don't exclude the possibility that the information might be transmitted through multiple polysynaptic pathways in the network. And again, as I was mentioning, we would like to study this communication model of polysynaptic relay information transmission by using both the structural connectivity information and the functional data. So we don't want to just, let's say, trace structural polysynaptic pathway, but we want to use the functional data to assess how much those structural pathways are compatible with information transmission along that path. So let me know how we proceed and how we decided to build our model. So let's consider a series of brain regions, here represented by this gray ball, and then a polysynaptic structural path between the source and the target region. So those green lines represent the white matter bundle. And the polysynaptic pathway is simple because we are not looking at direct connection between source and target, but at indirect connection that pass through other brain region. Then we can associate it to each brain region a time series recorded, for example, with functional MRI. And then we can see each one of these time series as the realization of a random variable. Here, for example, we have those three random variables x, y, z, each one associated with a probability density function. And in information theory term, we can quantify the amount of information shared between a pair of brain regions by computing the mutual information between those region pair, right? So this is a relationship between their probability mass function. Now, one of the fundamental principles of information theory is the data processing inequality. So these principles say that if three random variables form a Markov chain, x, y, z, then there is no processing at y that can increase the information that z contain about x. This translates in the fact that the pairwise mutual information between the first node and the following node cannot increase along the path. So, said in more simple terms, it's a bit like the telephone game we used to play when we were kids. So the first person in the line has a message and he wants to transmit the message to the last person in the line. And the fun part of the game is that the message can be received at the end of the line unaltered. So hello will be hello, but most often there is a sort of degradation of the signal. The message gets changed along the chain, so that in the end we have a funny message coming up. So this means that information, if we just think in a simple relay of information through an independent step along a polysynaptic structural path, information about the starting node X available and the last node Z, so information about the starting message available at the end of the chain, cannot increase along this transmission process. It can just be transmitted unaltered, or in the best scenario, or it can get degraded along the way. And since the Markov chain doesn't include any directionality information, the data processing inequality also stated that the pairwise mutual information with respect to the target node, so in the opposite direction of the path, also cannot increase, right? So this relationship should be valid when navigating the path in both directions. So we decided to use mutual information value along a structural path and the data processing inequality as a proxy of relay communication along this polysynaptic communication pathway. So what we do, we select a series of brain regions, and then we check the pairwise mutual information value along the path in the two directions. And if the mutual information value along the path are not increasing, then we deem that polysynaptic pathway as a possible channel for relay of information. If, on the contrary, along the path there is an increase somewhere of mutual information, then that pathway is not compatible with the idea of relay of information from between the two ending nodes, from the source to the target. So how do we operationalize this concept? So this is the methodological framework that we develop. So from one side, we have the structural connectivity network, the structural connectome, for example, derived from diffusion MRI or from track tracing. And then for each pair of brain region, I and J, we lay down some convenient structural paths. So these are short paths, so including a limited number of connections along the path between region pair I and J. In literature, this is also sometimes called the K shortest path, because we consider the first shortest path between two brain regions, the second, and so on. And then at this point, we plug in subject-specific functional information in the form of this mutual information value from, in this case, fMRI recordings. And we, let's say, check every structural path by looking at the time series along the path with respect to the data processing inequality. If the mutual information value along a specific path are not increasing, then we label that the structural pathway as a relay communication channel for relay information transmission from I to J. If, on the contrary, mutual information is, let's say, increasing along the path, then that path is not compatible with the idea of a relay of information in information theory terms. And that says that then we can build the different kind of measure from this concept. For example, a simple things that we can do is simply to count the number of structural path or the percentage of structural path that respect the data processing inequality. So we get a parallel communication score, where for each region, we can identify the amount of parallel communication taking place between the two regions. Let's say we can have a large parallel communication score, meaning that many structural paths respect the data processing inequality. And therefore, between region I and region J, we have a communication paradigm tailored toward parallel processing. On the other side, we can have other pair of region, I and J, with maybe no or just one structural path respect to the data processing inequality. And then between those regions, we might have no communication, so parallel communication score equals 0, or a communication paradigm tailored toward selective or hierarchical information transmission. Well, we apply this framework to different data sets. So from a data set from the Human Connectome Process project of 100 healthy adults. And then we apply the same framework to data from macaque and mice, which were scanned awake. So this is a group of 10 macaque and a group of 9 mice scanned awake in resting condition, of course. We also contrasted the results on empirical data with different null models. So one possibility was to substitute the functional time series with just Gaussian uncorrelated noise. This is the case of absent communication. So all the signals there are noise. Another possibility is to shuffle the time series between the different nodes. So the time series are not modified, they are just shuffled across node. The structural connectivity information is not changed. So this null model preserves all the multivariate properties of the fMRI recordings, as well as the structural connectome. A third possibility is to shuffle the mutual information value, and again, the structural connectivity matrix is left unchanged, and then apply some smoothing of the mutual information value according to the structural connectome. This is the case, this represents the case of physiologically structured noise with respect to the underlying brain connectivity network. So it's noise which is smooth, let's say, or has some correlation between regions which are structurally connected. And then for the different model, we can compute again the parallel communication score matrices, right? Okay, so after building all this machinery, what could we observe? So our first results was that relay communication is not limited to the shortest path. So there are some network level communication model that test the possibility that communication between two regions in the brain pass exclusively through the shortest path, so the path with the smallest number of steps along the path. And we saw that this was not true. So here on the left, each dot represents a subject, and here we have the overall amount of communication pathway. So the pathway that do respect the data processing inequality when focusing only on the first shortest path between region pair, and when focusing on longer path between region pair. And in gray, we have a null model. So we see that indeed we do have a polysynaptic pathway compatible with relay of information, not only limited to the first shortest path. Here on the right, you have the group average parallel communication matrix, and here the histogram of this matrix. So we see that there are several region pairs that have a parallel communication regime with a score which is larger than 1 and a median value which is of 1.53. Now we computed the same quantity for the macaque and the mouse, and what we saw that also for these other species, relay communication was not limited to the first shortest path as seen here. But an interesting aspect is that for macaque and mice, we missed the right-hand tail of parallel communication score value, meaning that in macaque and mice, communication is more tailored towards selective rather than parallel information transmission regime. And so we could say that there is a communication gap between human and non-human primate. Human and non-human mammals. Of course, the mice are not primates. Okay, and this gap was present also when contrasting with the null model that I described before, and also when investigating different data sets. So we also look at data set, a replication data set for macaque and mice, which were anesthetized. Unfortunately, it's not so easy to have many unanesthetized awake animal data set, but let's say the communication gap between human and non-human mammals was present irrespectively of the anesthesia status. Okay, then we explore again the spatial heterogeneity of the structure-function relationship and communication paradigm across the cortex. And what we observed that in human, as in macaque and in mice, we did observe selective communication, specifically in mainly in let's say unimodal and multimodal areas, so visual, somatomotor, and attention network. But in human, not also the different scale, color scale for the different species, we also observed larger parallel communication value, and so more parallel communication in a region which are part of the frontoparietal executive control network, and in the precuneus, which is the hub of the default mode network. So communication architecture relates to brain functional organization, we could say partially in different way in the different species. And then here, in the same vein, we investigated parallel communication within and between different systems. So within unimodal system, visual and attention network in the 3 species, transmodal system, control, default mode network, and limbic system. And then between these different systems, we call this cross-modal communication. And we observed that in human, but not in macaque and mice, there were many parallel communication streams connecting unimodal and transmodal region, as seen here. This was true both at the group level and at the subject level. And here again, I'm showing you the results when considering different replication data set. Here again, each dot represents an individual subject, and I'm showing you here the average parallel communication score between these cross-modal regions, so unimodal and multimodal region. We see how is higher in human compared to the non-human mammals. And I think that these results was interesting because it somehow complements some hypotheses that have been put forward in the 90s by Mars and Mesulam, who postulated this sensory-fugal axis, trying to explain how more abstract texture of cognition can emerge in human subject. So he postulated or hypothesized the existence of this axis from unimodal region with a more, let's say hierarchical connectivity to transmodal region here that have more interconnection, let's say more possible parallel stream of communication from unimodal to transmodal region. So in a way, these results let's say give a confirmatory perspective, let's say, of this hypothesis. Okay, and this is the last empirical results I would like to show you. We played Guess Who games also with the communication pattern. So we try to identify a subject from test-retest data, not only human, but also in macaque and mice. And the nice thing was that we saw that it is possible to identify a subject from a very simple measure of communication, which is this parallel communication score, a very highly compressed measure of brain communication process. So it was possible to identify individual subjects irrespectively of the species, so for all species. But what we also observe is that when we focus only on the brain region with on the brain system with low parallel communication score, so for more hierarchical selective information processing, or the one with high parallel communication score, so the one with more parallel and broadcasting information processing, we saw that the latter were the one that allowed a better subject identification, so that they were more subject-specific. In a way, in line with the results we saw about the structure function decoupling and the fact that it is the decoupled part of the functional activity pattern, which are more specific to individual subjects. Just a few considerations and limitations, let's say, of this work. So here we really try to put together functional and structural data to better understand the communication paradigms in brain networks. But of course, this is a really simple modeling of what may go on in the brain, right? We just consider simple information transmission between region and traveling across polysynaptic pathway, but we are not considering more complex communication mechanism. For example, we are not considering any kind of processing or nonlinear processing of information or more complex communication mechanism, for example, using some combinatorial approach or some non-Markovian approach where the steps along a path are not statistically independent. So there is a lot more work to do in this direction, I think. So to conclude, these two approaches and embedding of structural and functional information told us that communication protocols are heterogeneous across different brain networks, and in particular, we observed the selective hierarchical information transmission in unimodal areas, which are also strongly coupled with the underlying structural connectivity architecture, while we saw a more parallel or broadcasting transmission streams connecting lower order and higher order and transmodal regions in human. We saw, concerning the heterogeneity across individual, we saw the relevance of the part of functional activity pattern which is decoupled from the structural network, and we also observed that parallel communication regime is the one that is more specific to individual compared to hierarchical communication regime. And then finally, concerning the heterogeneity across species, we saw that some aspects are preserved, this is selective information transmission from unimodal area. But we also saw a parallel communication gap between humans, more tailored toward parallel communication, and non-human mammals. So in terms of outlook, of course, there are really many possible ways to go. I already mentioned the idea of integrating other aspects in communication models, such as the modification and the processing of information. And then, as Geisen was saying at the beginning, I'm working in a memory clinic in a hospital. So we are very much interested in extending this approach to clinical, or let's say cognitive setting, for example, to understand brain plasticity mechanism, how brain function might reorganize after a lesion or after a treatment. And then is this parallel communication somehow a proxy of brain reserve, let's say in cognitive, in neurodegenerative processes? And also it would be interesting to apply this model to the study of phenomena which live and developed on the brain network and are partially driven by brain function, such as the spreading of protein in several proteinopathies, again, neurodegenerative disorder. And with this, it's all. I would like to thank you for your attention, and thanks all of the people and collaborators involved in this work. So the MIP Lab, led by Professor Dimitri Van de Ville, and my colleague there, Enrico Amico, and Maria Giulia Preti. And then Gilles Alali, who is the head of the memory clinic and the SHUV, so the Lausanne University Hospital, and all colleagues and collaborators of the last year. And Kaisen for meeting you last year and for catching up so nicely.